Alrighty. Hello everybody. Apologies for how late this video is. This video is obviously not going to be very useful for those of you who've already who've already finished 441, because the class finished like on Monday of this week. So yeah, this is more so for the folks that are taking biochem in future years, but I wanted to make sure I got through all the lecture slides instead of finishing like halfway through and just calling it quits. So today we're talking about amino acid oxidation and the urea cycle. And yeah, let's go ahead and get into this. So amino acids, there's a couple reasons where you want to oxidize or degrade them. For example, maybe if you have too many amino acids, right, you can digest them to create sugars and all that stuff. So yeah, basically if the ingested amino acid exceeds the body's need, and this can occur if you have high protein diets, things along those lines. Yeah, these proteins can be used as like fuel sources, so they can be broken down, they can be turned into amino acids. These amino acids can be fed into the Krebs cycle or other intermediates to actually generate glucose or generate reducing agents to use in electron transport chain and such. So essentially, amino acids can be broken down and used to make electron carriers, which can be used to make ATP. Period. So, and here we're talking about just what the different pathways. So you can have intracellular proteins, dietary proteins. These can be broken down to make amino acids. Notice that you can use amino acids to make intracellular proteins, but then these amino acids can be used to make like I said, proteins and other nitrogen-containing molecules like neurotransmitters and nucleotides, stuff like that. And they can also just be oxidized and degraded and turned into ATP. So here we have the carbon skeleton. They can funnel into citric acid cycle intermediates. This is like fine. I don't think this is anything too crazy. We kind of already talked about this. Also, this is a cool little set of facts to know, I suppose. But the main thing that you should know from this is the amino acids. They can be broken down and turned into energy, so they can be used to make energy. They can be some of the amino acids can be turned into to citric acid cycle intermediates, so you can do gluconeogenesis with them. You can also turn some of them into like fatty acid prerequisites and cholesterol prerequisites, and you can also turn them into ketone bodies, certain ones into ketone bodies. So all this to say that you can use them to make energy again. So one of the things that is kind of cool to take a look at is glutamine synthetase, which is the creation of glutamine, which is the amino acid. And this comes about for a variety of reasons, but one of the main reasons is if you've got free ammonia produced in your tissues, right, and so nitrogen is a relatively toxic compound to your body. You don't want too much free nitrogen floating around in your body. You can combine it with glutamate via this synthetase to make glutamine, and this specifically occurs in the cytosol of the liver, kidney, and the brain. It does not occur anywhere else, so keep that in mind. And what's important about this enzyme, glutamine synthetase, is that this is how we transfer nitrogen groups, amino groups, in the bloodstream to other tissues so that we can use it for biosynthesis. And also, it's also used as a carrier of amino groups to the liver so that you can excrete it in the urea cycle. So basically, if you want to secrete urine, you also use glutamine to carry some amine groups that are excess, and you dump those into the urea cycle, and then you can release some ammonia. So here's a nice little schematic of what that enzyme looks like. We got glutamine here. Well, that's actually not glutamine. It's actually a poor diagram. This is glutamate, not glutamine. This is glutamate. Glutamine synthetase, what's going to happen is the intermediate is going to first have an ATP used, because remember, it's a synthetase, so it uses ATP. We're going to use ATP to activate this carboxylic acid group, and then we're going to use this amine ammonium as like a nucleophile, if you will. And then this is going to basically attack carbonyl and kick this off. So you end up with glutamine. And then when you get to the liver and you want to release the ammonium and use it in the urea cycle to release your ammonium in the urine, you use the enzyme called glutaminase, and this enzyme is specifically in the mitochondria of the liver. And what that does is that cuts this bond here. And when it cuts that bond, the ammonium basically leaves and goes into make urea, like we said here. So here's a bit more about glutaminase. So glutaminase is in the mitochondrial matrix, specifically of the liver. And so what happens when glutamine enters the matrix, it does so through some specific transport proteins, but I don't think that's necessarily important for this class. But this enzyme glutaminase is also present within the mitochondrial matrix of the liver, kidney, and brain. So basically the same as glutamine synthetase, except for this is in the cytosol, while this is in the mitochondrial matrix. So it's a bit of a different thing. And this is just like a hydrolase enzyme. It uses water to release ammonium. And here's that reaction. You use this water attacks this carbonyl and kicks this group off, basically. And so you release the ammonium and you end up with this O group again. That's what glutaminase does. Glutaminase does not use any energy, no ATP required. So, how does glutamate enter the mitochondrial matrix? We talked about this already. It enters through some transport proteins. Again, I don't think it's that important for the class. If they cover it later, I'll talk about it, but I don't think it's important for the class. But the main thing you need to keep in mind is this other enzyme, glutamate dehydrogenase, which is also within the matrix of the liver, kidney, and the brain. And this is important because this converts glutamate into alpha-ketoglutarate. So it's a dehydrogenase reaction, so it basically uses NAD+ or NADP+, and it oxidizes glutamate into alpha-ketoglutarate. And so it's a two-step process, basically, and in the end it releases the ammonium group that's here. So it's like, if you have a glutamine, what you do is you first use glutaminase to remove the ammonium group here, and then you use glutamate dehydrogenase to remove this group. In similar fashion, you can also use glutamate dehydrogenase to make glutamate from alpha-ketoglutarate. So it's a bidirectional, it's in equilibrium, or it's a reversible reaction, I should say, specifically. What else do I want to mention about that? But yeah, like I said before, used for biosynthesis, so creation of these amino acids, or excretion of the ammonium via the urea cycle. So either one. So why is ammonia toxic? And the reason it's toxic is because it's a base, actually. So it actually rips protons off of water and creates OH-, which actually raises the pH of your blood. And you should know this by now, hopefully, that your blood is very tightly regulated for its pH. It's around 7.2-ish, if I'm not mistaken. And so if you deviate from that, even going to 7.4 or 7.5 is already quite dangerous. But you can imagine if I add a bunch of hydroxide, my pH jumps to it can jump to really high numbers, and that's really bad for a lot of the enzymes in our bodies. A lot of the cells require certain pHs to function properly. So you can imagine how this can be detrimental to the cell and to the enzymes within those cells. So that's the idea. Basically, if you have this ammonia, you can react with water, forms this ammonium hydroxide species, and this dissociates as a salt to ammonium and hydroxide, which messes up ions, and it's messes up aquaporins, it causes cognitive impairment, all sorts of issues. So basically, the main thing you should get from this is that there is a significant need to transport excess amines to the liver in order to turn that into a non-toxic group. So yeah, and that's basically why we use glutamine and alanine. So glutamine is a way of transporting these amine groups in a non-toxic form, and same with alanine. And so yeah, ammonia deposited in the liver to enter the urea cycle for excretion. But yeah, like we talked about before, these two things here are carriers of ammonia, and these carriers are used to put these ammonia in the liver so that you can excrete it through in the form of urine. So this is a nice little pathway actually of like what is actually happening. So there's two kind of main things to talk about here. Are you talking about muscle or are you talking about tissue? Most other tissues, essentially. So there's two mechanisms to transport ammonia to the liver. And for most tissues, it is transported in the form of glutamine. And this is nice because it basically has two different amine groups attached to it, right? So glutamine, alpha-ketoglutarate first. You attach one ammonia to it, it becomes glutamate. Then you attach another, it becomes glutamine. So basically, it carries two amine groups for the price of one carbon skeleton, if you will. So we use glutamine synthetase and glutaminase in the liver, right? So we use glutamine synthetase in these other tissues, and then when we get into the liver, we use glutaminase to release it. In muscles, however, we actually form alanine instead, and then we use an alanine aminotransferase to basically take a pyruvate and a glutamate, and we switch the carboxyl group of a pyruvate, or I guess we switch one of the carbonyls of a pyruvate with the ammonia of a glutamate to make alanine. So if you see that it's a transamination reaction, you can see here this glutamate becomes alpha-ketoglutarate. So notice how it loses that ammonia group, and then pyruvate becomes alanine. So it's actually gained a ammonia group there. So that's basically the main idea. And this should hopefully make sense because muscles are constantly contracting. So they're always using glucose. They're always making pyruvate. So pyruvate is actually very common for these muscle cells. That's kind of the main thing to keep in mind. So now if we look at the metabolic fates of these ammonia, we'll talk about this real quick. The glutamine, once it has gotten into the blood and eventually into the liver, we use glutaminase to break it down into glutamate, and then we release one of the ammonias, right? And then once we turn it into glutamate, we then use glutamate dehydrogenase to release another ammonia. So we've actually released two different ammonias in this process. Like I said before, that's an important thing to keep in mind. Similarly, once from the muscle cells, once we've taken this alanine and we've put the alanine into the cell or into the liver, we use alanine aminotransferase again to form pyruvate, which we can then use in gluconeogenesis in the liver to make glucose, and we can ship this glucose off to the muscles again because they need glucose. So this is like a nice little interesting like back and forth that the muscles have with the liver. Notice how the muscle uses glucose to make pyruvate, that pyruvate's turned into alanine, and then that goes back to the liver. And then how the liver does this really interesting process where it takes the alanine, uses the aminotransferase, turns it back into pyruvate, and then sends this glucose back to the liver, or back to the muscle, sorry. So that's always I always found that really interesting and how that mechanism works. And then also, of course, we use alanine and alpha-ketoglutarate to make this glutamate, and then this glutamate again is used through glutamate dehydrogenase to make ammonia. And also remember that this reaction going from alpha-ketoglutarate to glutamate also uses glutamate dehydrogenase. That enzyme goes in both directions. So, what is the fate of ammonia? There's a couple fates of ammonia. One of the first things that could happen is that once ammonia is deposited into the liver to enter the urea cycle, it could be excreted by the kidneys as urea. And so this compound contains two ammonium groups per one urea. So that's one option. The other option is that it could just remain as ammonium, and small amounts of ammonium is fine. You just can't have massive amounts. And the other option is that it's turned into uric acid. And we've talked about this before, but with uric acid, there's 4 different ammonia groups that are used to make it. So that's another fate that you could have. And this diagram just once again shows, what's going on here exactly. So Again, if we have glutamine, we use glutaminase to make it into glutamate, and then we can use the glutamate dehydrogenase to remove this ammonium. This is just a fun little interesting diagram. You can see that it works for other proteins as well, or other amino acids as well. So it works for alanine, like we talked about. It also works for just any other amino acid that ingested from a protein. There's always going to be some ammonium group on them, so that you can always remove that with a transamination reaction to make some sort of alpha keto acid, and then a free ammonium group, which is then attached to glutamate. So basically, the way that I'm trying to say that a lot of different amino acids can donate their ammonium groups to alpha ketoglutarate to make glutamate, but yeah. Ultimately, the main thing that happens is you make ammonium, urea, uric acid, and that's excreted in the urine usually. So, like I talked about, how is ammonium generated? So ammonium is generated in the mitochondria of the liver, kidney, and the brain through these two enzymes, glutaminase and glutamate dehydrogenase. That's two different ways of making ammonium. And so the other thing we also need is we need a couple of things. First we need our ammonium. So if we're talking about the urea cycle now, by the way. In the mitochondria, right, we need two things. We need an ammonium and we need a carbon dioxide. Now, remember, how do we get carbon dioxide usually? We can't just pluck it out of thin air. So we use bicarbonate. Remember, HCO3 minus. That's a horrible drawing of HCO3 minus. I hope you understand what I'm saying. HCO3 minus. Bicarbonate is the main way that we get CO2. So remember that's the main thing that we use. So we use this ammonium and CO2 to make this thing called carbonyl phosphate. And along with that, we use ATP with the CO2 and the ammonium to make this carbonyl phosphate. This is made within the mitochondria, and it's done through this enzyme called CPS1, carbonyl phosphate synthetase 1. And like I said, ATP-dependent reaction. That is the thing that we see up here. Basically what we're doing is we are taking this bicarb, we are attaching an ammonium to it, and then we're activating it. And notice how there's two ATPs being used. I think the way that I would think about this is I would think about it like one of these ATPs is used to activate one side of this so that this ammonium can attack, and the other side is used to activate the opposite side. So you can see here, one ATP used to activate this side so the ammonium can attach, the other side used to activate this other side, so we ultimately make this thing. So that's the main thing to keep in mind. Once we have made our carbonyl phosphate, we can attach it to our ornithine to make citrulline and all that stuff. But yeah, hopefully you're able to see how, we're using this ATP and these phosphates to kind of activate this molecule. And the reason that only one phosphate is released, right, is because the other one is still attached to the carbonyl phosphate. So, yeah. Here is actually exactly what I was talking about. This is the mechanism of carbonyl phosphate, the CPS1. So you can see here how we have this. Notice how it activates this and it grabs it. So we have this now, right? This ammonia group now attacks there to kick off that group leaves, this phosphate leaves. So now we have this. And then now we attack, now this basically grabs this, and yeah. Just realize that this image looks a little bit different from what it looks like up here. Don't think it matters too much, I'm going to be honest with you. It shouldn't make too much of a difference, but notice how it is a little bit different. So it doesn't exactly look the same. But I think either way it's okay. You can think of it as like either this side attacks or this side attacks. It doesn't really matter too much. I don't think they usually test on the structure specifically. Usually when they test on it, it looks like this though, if I'm not mistaken. Not like this. But don't quote me on that. Keep both of them in the back of your mind. Okay, so now we're talking about the first two steps of this cycle. So first, carbophosphate, remember, the carbonyl donate, a group donated to the ornithine, right? So this ornithine basically comes into the mitochondria through this transporter from the cytosol to the mitochondria. So we have this ornithine, we have this carbophosphate we just made. What we're going to do is we're going to take this nitrogen here, and we're going to use it to attack this. Or actually, more specifically, we should be taking, let's see here. Actually, no, yeah. We're going to take this, attack here, kick off this phosphate group. And so what we do then is we've made citrulline. So now we've made citrulline. Hopefully you can see how the nucleophilic attack works. This acts as a nucleophile. And remember, like this, like the plus-minus charge stuff, that is subject to change in the body. Like all these enzymes, right? They can make things basic and deprotonate, all that stuff. So that's something to keep in mind. That's why this thing works. So this enzyme is called ornithine transcarbamylase, and hopefully that makes sense. It is using ornithine, and it's a transcarbamylase. This is a carbamamide, I think? Carbamide? I can't remember the exact name. But it's like it's an amide, basically. And so that's where it gets its name from. It's a transcarbamylase. So it switches out this group, basically, and attacks this guy to create this. So we got ornithine transcarbamylase, and we lose this phosphate in the process, the inorganic phosphate. Then we have this transporter that takes citrulline from the matrix into the cytoplasm, and this is called an ornithine transporter, ORNT1. And so what that does is for every citrulline it pulls out into the cytosol, it pulls one ornithine into the mitochondrial matrix. So it's basically an antiporter, if you will. So one goes in, one goes out. So once we have our citrulline, we then, in the cytosol specifically, we then have this other enzyme called arginosuccinate synthetase. And so what this does is this step here is actually used to provide the second ammonium for the eventual product, which is urea. So if we look here right now, we can see this aspartate, right, is a not cofactor. It's actually a reactant in this whole thing. So we need an ATP molecule, and we also need an aspartate. And so what you're going to actually do is you're actually going to do a bit of an interesting reaction where the electrons on this ammonium come, basically, this ATP, right? We use this ATP to attach a phosphate to this nitrogen, if I'm not mistaken. That's the way you can think about it. We use it to attach a phosphate to this nitrogen. And then what we do is we resonate these electrons down, essentially. And these electrons from this, this double bond basically attack the place where that phosphate was and kick it off. And so what you end up with is this, like double bond thing shown here, which is arginosuccinate. And so this is favorable because one, we use one of the phosphates to activate this group, and two, we hydrolyze the pyrophosphate that is left over. So that's the main thing I would keep in mind. So like basically this specifically the AMP group basically attaches here, and this pyrophosphate is hydrolyzed. So we eventually hydrolyze the AMP off of this group, and the pyrophosphate is also cleaved in half to release a bunch of energy. Okay, so once we have arginosuccinate, what we then do is we use an enzyme called arginosuccinase. And this is very similar to like fumarase in a way where it I can't remember what fumarase does anymore. Like adds the double bond, if I'm not mistaken, to succinate. And so all we're doing now is we are literally taking this group here and we are cleaving it. So basically these electrons here basically come back and they form a double bond there. That's pretty much all you need to know. So once that's done, we've released fumarate as a byproduct, and now we have arginine, which is just an amino acid that you guys know and love. And so the difference between arginine and citrulline, I hope you can see, is this one singular carbonyl group. In citrulline, it should not be a carbonyl group, right? So arginine has all three of these being ammonium groups or nitrogens, while citrulline has two ammonium groups and then an O. But then the final step in this whole process comes from the enzyme arginase, and all this enzyme does is that it uses water to attach a oxygen on here instead, and so it's basically a cleavage reaction where this O actually attacks here, kicks this group off to form ornithine again, and then you do a little bit of resonance, and you'll get this product here, which is urea. And so then you can do the whole cycle again because you've now formed ornithine, and that ornithine is what is used to continue the cycle again. So yeah, again, enzyme requires H2O, and the arginine represents a precursor for ornithine. So all that to say that, you know, eventually this urea, once it's made in the liver and all that stuff, it's released into circulation because it's not as toxic as a free ammonium in the form of urea. And the reason it's not as toxic in the form of urea is because there's very strong resonance between both of these amine groups into the carbonyl. So basically, it can go like this, it can go like this, and so therefore it's not toxic anymore because there's really no potential for it to act as a base when it can resonate so strongly. And so yeah, urea is released into circulation and then it's excreted via the kidneys. And like I said before, the ornithine that's recreated is transported back to the mitochondria through ORNT1, and you can continue the cycle again. I won't harp on this that much. Just know that, you know, in this whole thing, right, we have to get aspartate. Aspartate is something we actually end up using in this cycle. So where do we get this aspartate from? We get this from the malate aspartate shuttle, which is something that you guys have already seen before in Biochem 440. And it's used in that class as a way of transferring NADH from the cytosol into the mitochondria so that the mitochondria is able to use NADH as an electron carrier for electron transport chain. But it's a bit different here. Now we don't care about the NADH. We care about the aspartate. That aspartate is what's useful here in this cycle. So, yeah, essentially a basic gist of this is, basic gist of this is the malate that we have in the mitochondria. We oxidize toxyl acetate. We then may turn it into aspartate, and then we shuttle that out into the cytosol. And then once it's in the cytosol, that aspartate can be used to in this cycle to turn citrulline into arginosuccinate. That's the main gist of it, though. And this is a fun little slide. Just keep in mind that these cycles operate independently. They don't have to. They're interconnected, yes, but they don't depend on each other. So yeah, that's kind of the main thing with this reaction, or this diagram. It kind of just shows everything, what's going on. So, yeah, so oxaloacetate, remember, can be converted into aspartate using aminotransferase reaction. And yeah. Basically, again, this whole thing is this. Aspartate formed in the mitochondria is transported into the cytosol via the malate aspartate shuttle to serve as an ammonium donor. So it can, that's the other thing. The aspartate doesn't have to go into this cycle necessarily, right? It could also be used as an ammonium donor, so you could use it to make ammonium, and then that could be used here instead. So there's a whole bunch of things that could happen, right? It's important to think about these in terms of inputs and outputs and not memorizing exactly. You should memorize, sure, like what goes in, what comes out, but you should also be mindful that, like, oh, where do I get this ammonium from? Oh, well, I can get it from multiple places. I can get it from glutamate, glutamine, aspartate, alanine. I can get it from multiple places, right? So that's kind of the main thing I would try and remember with this, is that these pathways are very interconnected. They don't always follow a straight line. So, yeah. Yeah, and so remember, right, so the fumarate, so the fumarate can also be turned into malate, because remember, you just have to use, I just realized, I think I made a mistake earlier. Fumarase is the one that adds a water to fumarate. Succinate dehydrogenase is the one that makes fumarate from succinate. So, my apologies. But, as I was saying, fumarate can also be turned into malate through fumarase, and then this is in the cytosol, remember? Cytosol. So you can add a water to this guy, and then you can, once you add a water to him, you can transport him to the mitochondria through the malate aspartate shuttle again, and then yeah, basically it's a reducing agent carrier at that point, right? So you can, yeah. So they're very interconnected, is what I'm saying. So basically, fumarate turns into malate. This malate can be oxidized by NAD+ to make NADH in the cytosol, and then this NADH is transported into the mitochondria via the malate aspartate shuttle, and then it's used in electron transport chain, right? Or fatty acid oxidation or whatever. So glycolysis, it's producing glycolysis, right? All of these things are really important to keep in mind. Basically, right, all that to say, fumarate to malate, malate turned into oxaloacetate, and then that NADH can be brought into the mitochondria to do electron transport chain. So that's the main thing here. Where is it? I'm trying to find it. Yeah, so like this, basically, right? So the malate, once it's made in the cytosol, goes in here, goes all the way around, basically, and basically comes in again, so. Right, so the malate can come in, and then it can be used to make NADH. So, regulation of it. How does regulation work? The main thing for regulation is the inactivation of CPS1. So CPS1 is inactive in the absence of N-acetylglutamate. So N-acetylglutamate is actually an activator of CPS1. So N-acetylglutamate is made through the combination of acetyl-CoA and glutamate. And this should hopefully make sense, because what we're signifying here is that we are in a high energy environment. We have a lot of acetyl-CoA, we have a lot of glutamate. That means that we probably have excess amine groups, and so we're going to have to get rid of them. So that's why it's triggered by acetyl-CoA and glutamate. And so what you get is you use this enzyme called N-acetylglutamate synthase, and that attaches these two to make N-acetylglutamate, releases CoA, and also it is activated by arginine. So if you have excess arginine, so excess proteins basically, and excess energy sources, it's turned on. And so you are more likely to do your urea cycle when you have excess proteins. This should just make sense, right? If you have excess energy, you're going to want to get rid of anything that's toxic to the body, and excess energy in this case means excess proteins. Excess proteins have a lot of ammonium groups. Those can be toxic if not dealt with. So yeah, once you make this activates the CPS1, like we talked about, and so then we make more carbamoyl phosphate, and then we make more urea. There are plenty of genetic defects in the urea cycle that can lead to ammonium toxicity. Break any of these enzymes, basically, and you are not able to get rid of your ammonium. And so you end up with a basic blood pH, and oh boy, not fun. So, yep. So now you're wondering, oh, okay, well, we've got rid of the ammonium, but what happens to the rest of the carbon skeleton? Well, it's a great question. Like we talked about, the ammonium groups transported to the liver in the form of glutamine and alanine, and then they're excreted in the urea cycle. But the carbon skeleton is actually put into things like citric acid cycle, so it's further oxidized in citric acid cycle to make ATP. In the liver, though, it can be used for two things. It can be used to oxidized to make ATP, or it can be used in biosynthesis, to gluconeogenesis, to make fatty acids, to make ketones, and that's where this next slide is important. But this slide here is just showing you that oxidation of carbon skeletons from amino acids accounts for 10% of the energy production, so it's actually a decent amount. And so it's especially common during conditions of like low glucose, low fat storage, so like basically depletion of all other storage. That's when you start using amino acids as your primary energy source. So if you're fasting, if you're starving, if you restricted on sugar, if you're doing a lot of exercise for a long time, and also if you have untreated type 1 diabetes. So yeah, basically the liver will start using these amino acids to make glucose, and transport that to extrahepatic tissue, so tissues other than liver cells. This is because certain cells like rely solely on glucose. Like the brain, for example, is one of those organs that like relies heavily on glucose. I don't think it relies solely on glucose. I could be wrong there, but it relies heavily on glucose, and so the liver ends up synthesizing, breaking down a lot of amino acids, turning them into glucose, and shifting or sending those out and shipping those out to the brain and other organs that rely entirely on glucose. The liver also synthesizes ketone bodies, and ketone bodies, remember, are just basically acetyl-CoAs combined together, and also transports these to extrahepatic tissue. And these are used as metabolic fuels, and apparently these are particularly used in the brain. So yeah, I think the main thing to keep in mind here is that these amino acid carbon skeletons are used as like that last ditch resort for energy. And so they can be used to make glucose molecules to send to some cells, and they can also be used to make ketone bodies. So yeah, this slide, all I'm going to say about this slide is you don't have to know every single bit of it. The ones that I would remember are for sure glutamine, glutamate, aspartate, asparagine, alanine, right? These are all gluconeogenic. So what is glucogenic, sorry. What does glucogenic mean? Glucogenic means that they can go into the citric acid cycle and they can be turned into oxaloacetate, and then remember, oxaloacetate can be turned back into pyruvate, and then you can use the pyruvate to go all the way through gluconeogenesis to make glucose. So if it goes into citric acid cycle and it ends up at oxaloacetate, it is going to be a glucogenic amino acid. So glutamate, glutamine, aspartate, asparagine, and alanine. Those are the main ones you should know for sure. Those can be turned into oxaloacetate, which can then be turned into pyruvate, which can then be turned into glucose through gluconeogenesis. For ketogenic amino acids are ones that are not glucogenic, and specifically they get turned into acetyl-CoA, and then the acetyl-CoA is used to make ketone bodies or fatty acids. So these are very different. The ones that you should know for sure are leucine and lysine. That is it. You don't need to know anything else. Leucine and lysine specifically, these are only used as ketogenic amino acids. They can only be used to make fatty acids and ketone bodies. They cannot be used to make glucose. They're not glucogenic. Everything else is generally like a mix of the two. There are a couple in here that are not. So some that are like really easy to remember are that like tryptophan, tyrosine, where is it? Yeah, so the things like tryptophan and phenylalanine, these are like really big amino acids, remember? So like their carbon chains can actually go into a bunch of things. So all that to say is like these really big ring amino acids, these big structured amino acids, those can be used as both glucogenic and ketogenic. So if you want to remember that, go for it. Don't think it's required. The main things you should remember for sure are ketogenic and glucogenic. And for ketogenic, you should remember that leucine and lysine are the two. And for glucogenic, you should remember just the ones that you've definitely seen before. So glutamate, glutamine, asparagine, aspartate, and alanine. But yeah, other than that, if you want to memorize phenylalanine, tryptophan, tyrosine, feel free. I don't think you actually need to memorize it. It should be kind of intuitive, right? These are big amino acids. They can go into both pathways perfectly fine. But yeah, other than that, that's actually going to be the end of this lecture video. Again, I apologize to those of you that have already finished your final exam. This is not very useful to you, but for those of you in the future, I hope this is a helpful video, and happy studying.